हादसे में खो गई पत्नी। न्यू दिल्ली रेलवे स्टेशन स्टम्पेड। अरे आजकल क्या हो रहा है ना, स्टम्पेड स्टम्पेड, हर जगह स्टम्पेड। Packaging. Bouquet. Chromatin condensation is observed but tangled. Chromatin condensation. Leptotin. Chromatin condensation is observed but tangled. Structure look like bouquet. Appears the arises from nuclear envelope. Appears the arises from nuclear envelope. Appears the arises from nuclear envelope but tangled. Structure look like bouquet. Zygote. The condensed chromosome starts pairing. Pairing is done between the two homologous chromosomes of non-sister chromatid. Pairing is done between two homologous chromosomes. Pairing is done with the help of protein complex or synaptonymal complex. The condensed chromosome starts pairing. Pairing is done between two homologous chromosomes. Okay. Synaptonymal complex. Paternal, maternal. Homologous chromosome. Two homologous chromosome. Gene is located at same locus. Gene is located at same locus. 23 pairs. Pairing is done with the help of protein complex or synaptonymal complex. Synaptonymal complex formed between non-sister chromatid or synaptonymal complex. Okay, protein complex. Pairing is done with the help of protein complex. Pairing is done. The condensed chromosome starts pairing. The condensed chromosome zygote. The condensed chromosome starts pairing. Pairing is done between the two homologous chromosomes, homologous non-sister chromatids. Okay, synaptonymal complex. Pairing is done with the help of protein complex called synaptonymal complex formed between non-sister chromatid. Synaptonymal complex is formed between non-sister chromatids. Synaptonymal complex is formed between non-sister chromatids of homologous chromosome. Formed between non-sister chromatids of homologous chromosome. The process of formation. The process of formation refers as the process of formation of synaptonymal complex referred as synapses. The process of formation of synaptonymal complex is referred as synapses. Result into bivalent pair of two chromosome or tetrad condition. result into bivalent pair of two chromosome or tetrad. Four chromatid. Physical appearance is still not visible. In zygote phase. 46 chromosome, 23 pair. 23 bivalent. 46 chromosome, 23 pair. 23 bivalent. Number of bivalent is equals to half the number of total chromosome in cell. Number of bivalent is equals to half into number of total chromosome in cell. 23 bivalent. Okay, number of bivalent is equals to half into number of total chromosome in cell. Half into total number of chromosome in the cell. 46 23 pair, 23 bivalent. Okay, each bivalent is equals to 4 chromatid. Each bivalent is equals to 4 chromatid. 1 2 3 4 pair of 2 chromosome. Chromatid लिखना था ना. Each bivalent is equals to 4 chromatid and 1 tetrad. Each bivalent is equals to 4 chromatid. 1 bivalent is equals to 4 chromatid. 1 2 3 4 4 chromatid is equals to 1 tetrad. 4 chromatid equals to 1 tetrad. 23 bivalent is equals to 23 tetrad. Into 4, 92 chromatids. Dyad, 2 sister chromatids. Tetrad, 4 sister chromatids. 2 sister chromatids. Dyad, tetrad, 4 sister chromatids. Tetrad. 1 tetrad is equals to 2 dyad. 1 tetrad equals to 2 dyad. Nucleus degrades only at the end of prophase and not initially degrades. Nucleus degrades only at the end of prophase and still nucleus degrades only at the end of prophase and not initially. Nucleus degrades only at the end of prophase and not initially. Cell nucleus. Nucleus degrades only at the end of prophase and not initially. Okay. Zygote. Digote. Packaging. Diplotin and dikinesis. Okay. 46 chromosome, 23 pair. 23 pair. Okay. 46 chromosome, 23 pair. 23 pair. 46 chromosome, 23 pair. 23 pair है ना. Bivalent is equals to bivalent ही ना. Number of bivalent is equals to half into number of total chromosome. That will be 23 again. 46 divided by 2. 23 bivalent. Each bivalent has 4 chromatid, 1 tetrad. Bivalent. Four chromatid, 1 tetrad. Bivalent is equals to tetrad बोल सकते हैं ना फिर तो. Bivalent. Dyad, 2 sister chromatids. Tetrad, 4 sister chromatids. 1 tetrad equals to, okay, bivalent. bivalent chromosome. Bivalent chromosomes. Bivalent chromosomes. वन हो गया. वन टू थ्री फोर. Four tetrad. Bivalent chromosome. Four tetrad. 1, 2, 3, 4. Okay. Four tetrad. One, two diad. 3, 4. This is triad. Chromatids. Bivalent chromosome. Four chromatids. Four chromatids. Four chromatids. Right? Four chromatids. One tetrad. भैया इसको फोर टेट्राड नहीं बोलते, फोर वन टेट्राड बोलते. वन टेट्राड. डायड. टू सिस्टर क्रोमैटिड्स. टेट्राड. कितना कंफ्यूजिंग है. नंबर ऑफ बाइवैलेंट क्रोमोसोम. बाइवैलेंट मतलब ये पूरा वाला bivalent chromosome. Number of bivalent is equals to half into total number of chromosomes. कोई बात नहीं. 1/2 half into total number of chromosomes रहेगा. वही bivalent वाला वो कहाँ है ना. Bivalent. Each bivalent has four chromatid, one tetrad. 1, 2, 3, 4. These are four chromatid. CHROMATID. Four chromatid, one tetrad, four chromatid, and one tetrad. Four chromatid and one tetrad. Okay. 4 chromatid and 1 tetrad. 23 bivalent is equals to 23 tetrad. 23 bivalent. 23 tetrad. Okay. 23 into 4. 23 into 4. 1 4 4 8 4 3 12. 12 go to 1 is away. 1 4 4 8 92. 92 chromatids. 92 chromatids. CHR chromatids. 92 chromatids. Dyad. 2 sister chromatid. This is 1 2 dyad or tetrad. This is dyad. 2 sister chromatids. 1 2 3 4. 1 2 3 4. This is triad. Tetrad. DETRAD. Tetrad. Triad की तो बात ही नहीं है. This is tetrad. This is tetrad. 4 sister chromatids in tetrad, 2 sister chromatids in dyad. 1 tetrad is equals to 2 dyad. 1 tetrad equals to 2 dyad. This is our bivalent chromosome. And this is our dyad. 2 sister chromatids. Pakitin. Indiplotin. Human oocytes duration from NCRT. Okay. 15 months or years. Pakitin. Tetrad physically appeared as chromosome. Pakitin tetrad physically appeared as chromosome. Shorter and thicker. Tetrad physically appear as chromosome. Shorter and thicker. Formation of synaptonemal complex ensures crossing over. Formation of synaptonemal complex in zygote helps in Formation of synaptonymal complex in zygote helps in crossing over. Zygote synaptonymal complex, synapses, okay. Ensures crossing over. Formation of synaptonymal complex ensures crossing over. Helps in recombination. Crossing over helps in recombination. Combination of new genes helps in recombination, that is combination of new genes. Crossing over. For crossing over, recombination nodules. For crossing over, recombination nodules are observed. For crossing over, recombination nodules are observed. Synaptonymal complex ensures recombination or crossing over, helps in recombination or crossing over. Combination of new gene. Recombination helps in getting the combination of new gene. Combination of new gene. For crossing over, recombination nodule. For crossing over, recombination nodules are observed. For crossing over, recombination nodules are observed at some site along synaptonymal complex. Along synaptonymal complex, there are recombination nodule. Recombination nodule. For crossing over, recombination nodule are observed at some site along synaptonymal complex. Initiate functioning is achieved by recombinase enzyme. Recombination nodule initiate functioning. Recombination nodule are observed at some site along synaptonymal complex. Initiate functioning is achieved. Initiate functioning is achieved by recombinase enzyme crossing over for exchange of segment. Tetrad physically appeared as chromosome. Tetrad physically appeared as chromosome. Shorter and thicker. Formation of synaptonymal complex ensures recombination or crossing over. Recombination or crossing over. Recombination or crossing over. Crossing over helps in recombination. Recombination, yeah. Formation of synaptonymal complex ensures crossing over, which helps in recombination. Combination of new genes, okay. For crossing over, recombination nodules are also present or visible, are observed. For crossing over, recombination nodules are also observed. Recombination nodules are also observed. Recombination nodules are also observed. Recombination nodules are observed in packaging. Recombination nodules are observed in packaging. At some site along synaptonymal complex, recombination nodule initiate functioning is achieved by recombinase enzyme. Which enzyme is used? Recombinase enzyme. recombinase enzyme, okay. एक तो यहाँ पर recombination होगा, crossing over which helps in recombination. Recombination nodule is formed, and the third one is the enzyme used is recombinase enzyme. हाँ इतना ही तो है, which initiate the function of recombination nodule. Initiate functioning is achieved. Synaptonymal complex physically appeared as chromosome. Shorter and formation synaptonymal complex ensures crossing over, helps in recombination. Combination of new genes. For crossing over, recombination module are observed. Recombination modules are observed. Recombination modules are observed. For crossing over, recombination modules are observed at some site along synaptonemal complex. Initiate functioning is achieved. Recombinase enzyme. Initiate functioning is achieved by recombinase enzyme. Crossing over for exchange of segment. Crossing over for exchange of segment. Crossing over for exchange of segment. Diplotin. The fourth stage. 1, 2, 3, 4. Okay. Diplotin. Desynapsis. Removal of synaptonemal complex. Desynapsis. Diplotin. Desynapsis. Removal of synaptonemal complex. Some site between non-sister chromatid remains attached more than one site. Longest phase of prophase I. Diplotin. Longest phase of prophase I. Longest phase of prophase I. Okay. Diplotin. Desynapsis. Which one is the longest phase of prophase I of meiosis? It is diplotin. Here, desynapsis occurs. Desynapsis. Removal of synaptonemal complex. Removal of synaptonemal complex. Some site between non-sister chromatids remains attached. Some site between non-sister chromatids remains attached more than one site. Okay. Desynapsis. Removal of synaptonemal complex. Some site between non-sister chromatids are attached more than one site. Desynapsis. Removal of synaptonemal complex. Some site between non-sister chromatids remains attached more than one site. Desynapsis. Removal of synaptonemal complex. Some site between non-sister chromatids remain attached more than one site. Longest phase of prophase I. Non-sister chromatid. Some site between non-sister chromatid remains attached. Some site between non-sister chromatid remains attached more than one site. Okay. Appears like X. X alphabet referred as chiasmata. Actual site of crossing over. Chiasmata are the actual site of crossing over. Okay. Desynapsis. Removal of synaptonemal complex. But there are certain sites in the there are certain sites between non-sister chromatids which remains attached. There are certain site of sister chromatids which remains attached, and there the actual recombination takes place, or crossing over. Actual site of recombination or crossing over. They are visible in X shape and they are referred to as chiasmata. Chiasmata favors crossover. Chiasmata favor crossover. Termination of chiasmata starts in diplotin. Termination of chiasmata starts in diplotin. Chiasmata favors crossover. Termination of chiasmata starts in diplotin. Dikinesis. Termination of chiasmata. Okay, starts in diplotin but it ends in dikinesis. Termination of chiasmata. Exchange segment of chromatid. Exchange the segment of chromatid. Homologous chromosome. Non-sister chromatids, right? Homologous chromosome. Non-sister chromatid of homologous chromosome. Non-sister chromatid of homologous chromosome. Termination of chiasmata, exchange segment of chromatid, nuclear membrane disintegrate, nucleolus disappears and Golgi body disappears. Golgi body and nucleolus disappears, nuclear membrane disintegrate, exchange segment of chromatid, termination of chiasmata, dikinesis. Okay, Metaphase I. Anaphase I, Telophase I, Prophase, Metaphase, Anaphase, and Telophase, right? Okay. Then we have meiosis too. Okay. Significance नहीं दिया है क्या? Significance of meiosis. हजुर हजुर
हेर नानीले बाबा खाएन कि
ए हजुर
यसो 10 बजेतिर म यो खाना ल्याइदिएको छु कि
ए हजुर
कुकुरमा यो पाकेको छ कि एक सिटी लगाइदिनु कि सिटी लाग्नु पर्छ हेरेर भने चाहिँ यो दिनु नि ल म राखिदिन्छु कि हुन्छ आइहाल्छु म 11 बजेतिर ल हुन्छ हजुर खै भाइ चाहिँ आइसक्नु बन्द गरे हुन्छ जाने कि म आइहाल्छु ए हजुर हुन्छ हुन्छ फोनको पासवर्ड हजुर 2025
From 10, I have to babysit again. Till then, let's study. I want to cover some portion. Eight 36 से start किया था. 9:30. Metaphase I. Alignment of bivalent form of homologous chromosome at metaphase plate. Alignment of bivalent form of homologous chromosome at metaphase plate. Alignment of bivalent chromosome of homologous chromosome at metaphase plate. Alignment of bivalent chromosome of homologous chromosome at metaphase plate. Okay, don't think about it. Metaphase plate. Alignment of bivalent मतलब कि वो ही ना 4 tetrad वाला 1 tetrad 4 chromatid. Alignment of bivalent form of homologous chromosome at metaphase plate, spindle, equator or equatorial plate. Metaphase plate, spindle, equator or equatorial plate. Alignment of bivalent form of homologous chromosome. Kinetochores attached, 2 different centromere. Two different centromeres. Kinetochores attached, 2 different centrioles लिखा होगा यहाँ पर centromere लिख दिया है मैंने. Row 1, row 2. 2 rows of metaphasic homologous chromosome. Okay, there are 2 rows of metaphasic homologous chromosome. Metaphase. 2 rows of metaphasic. The bivalent chromosome aligns on the equatorial plate, which is also known as spindle equator, right? Spindle equator or equatorial plate. The microtubules from the opposite pole of the spindle attached to the pair of homologous chromosome. The microtubules from the opposite pole of the spindle, poles of the spindle attached to the pair of homologous chromosome. इतना ही दिया है यहाँ पर. 2 row of metaphasic homologous chromosome. 2 rows of metaphasic homologous chromosome. kinetochore attached to two different centrioles होगा शायद, सेंट्रोमियर लिखा है ना, कोई बात नहीं, गलत. Two rows of metaphasic homologous chromosome. Two rows of metaphasic homologous chromosome. They are homologous chromosome. Metaphasic homologous chromosome. Okay. Anaphase I. Homologous. The separation of homologous chromosome. The separation of homologous chromosome. No centromere division is observed. The separation of homologous chromosome. No centromere division is observed, like in mitosis. Chromosome separation is observed. Chromosome separation is observed. Anaphase I. Actual reduction division phase. Actual reduction division phase, as half set of chromosome moves towards each pole. Anaphase I. The separation of homologous chromosome. No centromere division is observed. Chromosome separation is observed, but no centromere division. No centromere division. Chromosome separation. Homologous chromosome separation. Okay. 46 chromosome, 23 pair, 23 each. Anaphase I. Actual reduction division phase. Actual reduction division phase. Okay. Anaphase I. Actual reduction division phase. As a half set of chromosome moves towards each pole. Half set of chromosome moves towards each pole. There are total 46 chromosome, but half set of chromosomes move towards each pole. Okay. Anaphase I. Telophase I. Separated chromosomes at each cell. Who cares, bro? Who cares? Separated chromosome at each cell. Separated chromosome at each cell. Decondensation of chromosome observed, but not to the extent of telophase of mitosis. Okay, decondensation of chromosome is observed, but not to the extent of telophase of mitosis. Telophase of mitosis. Okay, reappearance of nuclear membrane and nucleolus, ER and Golgi bodies. Reappearance of nuclear membrane, nucleolus, ER and Golgi bodies. Cytokinesis. Division of the cell followed by karyokinesis. Division of cell followed by karyokinesis. Transition between meiosis I and meiosis II. Intermeiosis or interkinesis. In interkinesis, DNA replication is not observed. Centriole duplication is observed. Leave it. Transition between meiosis I and meiosis II. The transition between meiosis I and meiosis II. Intermeiosis. Intermeiosis or interkinesis. In interkinesis, DNA replication is not observed. Interkinesis, within DNA replication is not observed. Centriole duplication is observed. Okay, interkinesis. During interkinesis or intermeiosis. Intermeiosis or that is transition between meiosis I and meiosis II. Intermeiosis or interkinesis. Intermeiosis or interkinesis. DNA replication is not observed, but centriole duplication is observed. DNA replication is not observed, but centriole duplication is observed. Leptoten bouquet. BOQUA bouquet. Like chromosome. CHROMOSOM is chromosomes. It appears like they are just Okay, bouquet-like chromosome. It appears like they are formed from the nuclear membrane. Appears like they are formed from nuclear membrane. Nuclear membrane. MEMBRAN. Nuclear membrane. Okay. Nuclear membrane. Electrolyte chromatin condensation is about but tangled. Structure look like a bouquet. Structure but tangled. Okay, structure look like bouquet. Appears they arises from nuclear envelope. But tangled. Okay, structure look like bouquet. Appears they arises from nuclear envelope. Chromatin condensation is observed but tangled. Structure look like bouquet. Appears they arises from the nuclear envelope. हो गया. जाइगोटिन में तुम्हारा होगा, ये बहुत ही इंपॉर्टेंट है, साइनेप्टोनिमल कॉम्प्लेक्स देता है ना. Zygotin of prophase. Is it prophase I? Yeah. Zygotin of prophase I, right? Prophase I. Synapse between homologous chromosome. Okay. Non-sister chromatid of homologous chromosome is the correct word. Synaptonymal complex form. This is the protein complex. SY, synaptonymal protein. PROTEI ना, protein complex. कोई बात नहीं, कितना ही सोचेंगे. Synaptonymal complex. Okay, formed between non-sister chromatid of homologous chromosome. Pairing is done between two homologous chromosome. Okay. Pairing is done with the help of protein complex, synaptonymal complex. The process of formation of synaptonymal refers as synapses. मैंने लिखा तो है. Result into bivalent pair of chromosome. Pair of two chromosome. Or tetrad condition. Yeah, this is chromosome. 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 These are bivalent chromosome. Bivalent chromosome. CHROMOSE. Chromosomes. SOMES. Zomes. This is dyad / two sister chromatid. CHRO chromatid. Dyad or two sister chromatid. Hi. Four chromatid, one tetrad, dyad, two sister chromatid, dyad or two sister chromatid. Bivalent chromosome, one tetrad, lactoprotein, bouquet-like chromosome, dyad. Okay, appears like they are formed from nuclear membrane. Then we have zygotin, black protein, zygotin, paquitin, recombinase enzyme, right? recombinase ही था ना वो एंजाइम का नाम. recombinase enzyme correct, recombinase enzyme, packaging. इसको ना मैं राइट लगा देती हूँ. recombinase enzyme, packaging, recombinase enzyme, packaging एक वो होगा, फेज होगा, recombinase enzyme, ENZYME enzyme, recombination occurs crossing over, crossing over between two non-sister chromatid CHRO chromatid of homologous chromosome की non-sister chromatid, right? Crossing over. For crossing over, recombination nodule, okay, formation of synaptonemal complex. Tetrad physically appeared as chromosome. Packaging. Tetrad physically appeared as chromosome, shorter and thicker. Packaging. Tetrad physically appeared as sapni. Packaging. Tetrad physically appeared as chromosome, shorter and thicker. Formation of synaptonemal complex. Packaging. Tetrad physically appeared as chromosome, shorter and thicker. Formation of synaptonemal complex. Tetrad physically appeared as chromosome. आई क्या लिखा है यहाँ पर मैंने? Tetrad physically appeared as chromosome, shorter and thicker. Formation of synaptonemal complex. Packaging नहीं ना? Ensures formation of synaptonemal complex ensure crossing over. Mention किया है मैंने, हाँ. Helps in recombination. Okay, formation of recombination nodule. ये should recombination nodule. Formation of recombination. Combination. It's okay. Recombination nodule. Formation of recombination nodule are observed at some site along synaptonemal complex. Initiate functioning is achieved by recombinase. Enzyme. Leave it, bro. Leave it. नहीं मेरे दिमाग में आ रहा है, कोई बात नहीं, चला जा. Initiate functioning is achieved by recombinase enzyme. Okay, crossing over recombination. For crossing over, recombination nodules are observed. Crossing over for exchange of segment. Diplotin. Longest phase of Prophase I. It is the longest phase of Prophase I. Desynapses occur. Removal of synaptonemal complex. Occur in diplotin. What is the characteristics of this phase? Well, the formation of chiasmata. X-shaped alphabet referred as chiasmata. It is the actual site of chiasmata are the actual site of crossing over. Okay. Desynapsis, removal of synaptonemal complex. Some site between non-sister chromatids. Desynapsis, removal of synaptonemal complex. Some site between non-sister chromatids are still attached or remains attached. Sister chromatid remains attached. Desynapsis, removal of synaptonemal complex. Some site between non-sister chromatid remains attached. More than one site. Okay. Reappears like X alphabet. Referred as chiasmata. Termination of chiasmata starts in diplotin. Termination of chiasmatin starts in diplotin and completely done in dikinesis. Termination of chiasmata. Okay. Termination of chiasmata. Exchange of segment of chromatid. Exchange of segment of chromatid is appeared in dikinesis. Exchanged segment of chromatid. Exchange segment of chromatid. I'm kind of sleepy, but I will not sleep because I have to look after my nephew. Exchanged segment of chromatid. Termination of chiasmata. Exchange segment of chromatid. Exchanged segment of chromatid. Termination of chiasmata. Exchanged segment of chromatid. Exchanged segment of chromatid. Termination of chiasmata. Exchanged segment of chromatid. Nuclear membrane disappears. Nucleolus, ER and Golgi, everything disappears. Metaphase I. Chromosome moves towards metaphasic plate or equator plate or spindle plate. Equatorial plate or spindle plate. Okay. Kinetochore is attached with centromere. Kinetochore attached with centromere. सेंट्रोमियर नहीं होना था, ये सेंट्रीओल्स होना था, मैं कंफ्यूज हो गई हूँ. That to observe two rows of metaphase homologous chromosome, two rows of metaphasic homologous chromosome. Anaphase I. Ana means upstage. Okay. Centromere do not divide, but chromosome starts moving towards pole. The separation of homologous chromosome. Separation of homologous chromosome is seen in anaphase. Two important points. Separation of homologous chromosome. Centromere division is not observed. These are the 2 points. Chromosome separation of homologous chromosome is observed, but no centromere division. Anaphase I, actual reduction division phase as the actual reduction division phase is anaphase, since 46 pair of chromosome is distributed among two pole, 23 each. Actual reduction division phase, half set of chromosome moves towards each pole. Actual reduction division. Okay, anaphase. The separation of homologous chromosome. No centromere division is observed. हाँ, तब मैं रिविजन कर सकती हूँ शायद. The separation of homologous chromosome. No centromere observed. No centromere division is observed. कोई बात नहीं ब्रो. Telophase I. The separated chromosome condense. Decondensation of chromosome. Observed but not to the extent of telophase. Of mitosis. Telophase I. Separated chromosomes at each cell. Decondensation of chromosome. हाँ, सब लोग फेक है. Decondensation of chromosome observed but not to the extent of telophase of mitosis. Separated chromosomes at each cell. Decondensation of chromosome observed but not to the extent of telophase of mitosis. हो गया ये तो. Reappearance of nuclear membrane, nucleolus, ER and Golgi body. Reappearance of nuclear membrane, nucleolus, ER and Golgi bodies. Okay, cytokinesis. Division of the cell follows karyokinesis. Transition between meiosis I and meiosis II. Interkinesis or intermeiosis. Intermeiosis or interkinesis. Interkinesis or intermeiosis. Interkinesis. DNA replication is not observed, but centriole duplication is observed. Interkinesis. During interkinesis, DNA duplication is not observed, but centriole duplication is observed. Okay. मैं पहले वाला भी थोड़ा रिवाइज कर देती हूँ, उसके बाद फिर NCERT करेंगे. पता नहीं, लेकिन उसका नाम कोलिकाइसिन था, ऐसा ही कुछ था. Where is it? हाँ यहाँ पर है. Colchicine. It is colchicine. Okay. Colchicine. ये एक बार होने के बाद I think वो भी तो करना है. Cell cycle continuous. The continuous process of maintaining the cell with reference to chromosomal separation is referred as cell cycle. The continuous process of maintaining the cell with reference to chromosomal separation is referred as cell cycle. The cell cycle. The continuous process of maintaining the cell with reference to chromosomal separation is referred as cell cycle. The continuous process of maintaining the cell with reference to chromosomal separation is referred as cell cycle. Includes 1. Preparatory phase referred as interphase or resting phase. Preparatory for actual division take place. M referred as interphase. M phase includes actual division of the cell. Interphase accounts for more than 95% of complete cycle. Interphase accounts for more than around 95% of complete cycle, whereas M phase accounts for 5% of complete duration of cell cycle, less than 5%. Okay, more than 95%, more than 95%. The cell division proper lasts for only about an hour. Corresponding to the M phase starts with the nuclear division. Corresponding to the separation of daughter chromosome, karyokinesis, and usually ends with division of cytoplasm, cytokinesis. Cell is preparing for division. M phase 5%. Different cells have different duration. Human cell 24 hours. For division is 90 minutes. Minute is very important. Okay, 24 hours and 90 minutes. Okay. The continuity of cell division preparation represents cell cycle. The continuity of cell division preparation represents cell cycle. Cell cycle: interphase, preparatory phase, resting phase. Actual cell division not observed. Comprises of 3 stages: cell phases, G1 phase, gap 1, synthesis phase, and Z2 phase, gap 2 phase, synthesis, and gap 2 phase, gap 1, gap 2, and synthesis. G0 phase, quiescent stage. Okay. G1 phase, gap 1 phase, most of the cell organelles duplication. Okay. So G1 phase, gap 1, most of the cell organelles duplication. RNA and protein synthesis. DNA precursor. RNA and protein synthesis. Somatic cell, diploid cell. Somatic cells are diploid cell. I'm kind of sleepy. I have to make coffee. DNA precursor, nucleotide, amino acids, sugar synthesis. Longer the phase, slower will be the division. Longer the phase, slower will be the division. DNA precursor, nucleotide, amino acid, sugar synthesis. Longer the phase, slower will be the division. RNA and protein synthesis, cell organelles replication. Number of cell organelles, most of the cell organelles replication. Okay, ये G1 का है. S में होगा तुम्हारा DNA replication or duplication of DNA. Then we have duplication of DNA in inside nucleus. DNA replication or duplication of centrioles in cytoplasm. Duplication of centriole in cytoplasm. Centriole. Duplication of centriole in cytoplasm. Okay. Centriole. Histone protein are also synthesized. Histone protein is also synthesized in S phase. G1 phase, chromosome X. Okay, if there is X number of chromosome in G1, after S phase, the number of chromosome will remain same, X. Okay, DNA duplication, sister chromatid duplicate. DNA duplication, sister chromatid duplicate. Number of chromatid increases. DNA duplicate, sister chromatid duplicate. Okay, if amount of DNA is in G1 is 2C, amount of DNA at S will be 4C. Example, if 8 pz at Z1, the amount of DNA will be 8. It will 16 pz at S. Okay, 16 pz. If the total number of chromosome at Z1 is X, okay, total number of chromosome will not change. Number of chromosome at S remains X. Okay, total number of chromosome will not change. Amount of DNA will obviously increase along with the number of chromatid. Number of chromatid at G1 is N, then the number of chromatid at S is 2N. Okay, G2 phase, gap 2 phase. Okay, first is, I don't think about it, Ima. Gap 2 phase, tubulin protein synthesis. Gap 2 phase, tubulin protein, microtubules, not tubulin protein. Tubulin protein synthesis, special set of organelle duplication, chloroplast, mitochondria, Golgi bodies. A special set of organelles replication, chloroplast, mitochondria, Golgi bodies. RNA and protein synthesis. RNA and protein synthesis, special set of organelles duplicate like mitochondria, chloroplast, and chloroplast, mitochondria, chloroplast, and Golgi body. Yeah. Tubulin protein synthesis. Tubulin protein is synthesized in gap 2 phase. Okay. Gap 1 of protein and RNA synthesized. That's it. Histone protein is synthesized in S phase. Histone protein is synthesized in S phase. Centriole duplication. DNA duplication nucleus. G0 phase, also referred to as quiescent phase. Referred as quiescent phase. Referred to as quiescent phase. Phase of metabolism in the cell without undergoing proliferation. Referred as quiescent phase. Phase of metabolism in the cell without undergoing proliferation. Without undergoing proliferation. Photosynthesis cell. Plant growth and development. G0 phase, referred as quiescent phase. Phase of metabolism in the G0 phase, referred as quiescent phase. Phase of metabolism in the cell without undergoing proliferation. Metabolism observed, cell proliferation not observed. The condition where cell can exit the cell cycle. The condition where the cell can exit the cell cycle temporarily in deficiency or requirement. It appears into action. Cell division. Permanently ceases to divide. Nerve cells permanently ceases to divide. Mitotic phase. Mitotic phase comprises of two processes. Mitotic phase comprises of two process, karyokinesis and cytokinesis. Division of nucleus and division of cell division. Nucleus division. Okay, karyokinesis, nucleus division. Cell regulation is regulated or maintained by certain signalings. Signaling refers to the surveillance mechanism of the cell to check, stop, maintain the different phases in the cell. To check or ensure the error-free process. S phase, G1, S, G0, M phase. Okay, M phase, prophase, metaphase, anaphase, and telophase. Then we have cytokinesis. Cell regulation is carried out by checkpoint surveillance mechanism. Okay, एक होगा G1S checkpoint, then the other. I don't think about it. G1S and the other one is G2M. The third one is anaphase promoting complex or spindle assembly checkpoint. Metaphase anaphase transition, metaphase anaphase transition, metaphase anaphase transition, anaphase checkpoint, G2M checkpoint, G1S checkpoint. 3 checkpoints. Okay, metaphase anaphase transition, metaphase to anaphase transition, metaphase anaphase transition, spindle assembly checkpoint, spindle assembly checkpoint, anaphase promoting complex, anaphase promoting complex. G1 checkpoint influenced by cell growth DNA synthesis, cell growth DNA synthesis. G1S checkpoint influenced by cell growth DNA synthesis, precursor component. Nutrient lack or non-regulation of precursor component, lack nutrients referred as a restriction point. G1S checkpoint is also known as restriction point. Once it is passed, cell cycle will complete. Once G1S checkpoint is passed, cell cycle will be completed. Restriction point. Okay. G2M checkpoint influenced by cell growth and DNA replication. APC influenced by microtubules formation. Microtubules formation. In the lack of formation, spindle assembly. Attachment of a spindle in kinetochore. No proper attachment. Checkpoints are regulated by cyclin protein, cyclin dependent kinase. Cyclin protein, cyclin dependent kinase or cell division cycle kinase. Cell division cycle kinase. Cyclin dependent kinase. CDK, CDC2 kinase. CDK, CDC2 kinase. Cyclin protein, CDC. Cyclin dependent kinase. Cell division cyclin kinase, CDC2, CDK. Kinase is activated when its joins attached to cyclin. Two types of cyclin are observed. G1 cyclin helps in G1S checkpoint associated with CDC2 kinase or CDK. Mitotic cyclin. Mitotic cyclin associated with CDC2 kinase, G2M checkpoint. Forms maturation promoting factor. Forms maturation or promoting factor. Mitosis. Observed by Starburger. Mitosis is observed by Starburger in plants, and in animals it is observed by Fleming. Term coined by Fleming. Okay, mitosis is the term coined by Fleming. Consists of two processes, karyokinesis and cytokinesis. Involves subphases. Cytokinesis involves two types of cell: animal cell formation, plant cell blade formation. Prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. Karyokinesis for subphases. Prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. Mitosis, division of one cell into two daughter cells. Each daughter cell receives equal number of chromosomes. Division of one cell into two daughter cell. Each daughter cell receives equal number of chromosomes, which is equal number of chromosome in parent cell. Okay, 46 chromosome parent ma chamani duita daughter ma pani 46 answer. Equational division, mitosis, generally observed in diploid cell in animals and both haploid and diploid cell in plant. Generally observed in diploid cell in animal and both haploid and diploid cell in plant. NN2N, generally observed in diploid cell in animal and both haploid and diploid cell in plant. Karyokinesis, one daughter nuclei is equals to two daughter karyokinesis. One daughter nuclei equals to two daughter nuclei in the same cell contained within the cell. Prophase, first stage initial event of prophase, chromatin chromosome condensation. Appears as entangled fiber centrioles, centromeres that was duplicated in as phase of interphase. Chromatin chromosome appears as entangled fiber centrioles that was duplicated in as phase of interphase starts moving towards the centrioles that was duplicated in as phase of interphase starts moving towards the pole. Appears as entangled fiber, chromatin chromosome condensation. Okay, matured centriole at Z2. Astral ray formation. Emergence is observed in the initial prophase. I can't think about it. It's okay. Astral ray formation, emergence is observed in the initial prophase. हम क्या कर सकते हैं वैसे? अमा astral ray formation, emergence is observed in the initial prophase. The chromatin fiber appears G2. Astral rays formation, emergence is observed in the initial prophase. The chromatin fiber appears like ball of wool, referred as spiring stays. Astral rays formation, emergence is observed in the initial prophase. The chromatin fibers appears like ball of wool, referred as spiring stays. Spiring stays. Astral rays formation, emergence is observed in the initial prophase. The chromatin fiber appears like ball of wool, referred as spiring stays. Based on astral formation, two types of mitosis. Without astral formation, anastral implant, centriole absent, anastral. Okay, without astral formation, anastral implant, centriole absent. With astral formation, it's okay. Astral, amphiastral, centriole present. Aster is composed of centrosomes with astral rays, and aster is composed of centrosomes with astral rays. In total, two asters, the two asters associated with the spindle fiber microtubules, collectively referred as mitotic apparatus. Mitotic apparatus, late event of prophase, chromosomal condensation, centriole movement polewards. The astral rays formation, late events of prophase, chromosome condensation, centriole movement polewards. Chromosome condensation, centriole movement poleward. The astral rays formation, the disappearance of nucleolus, Golgi bodies, the disintegration of nucleus, centriole movement polewards. Chromosome condensation, centriole movement polewards, the astral ray formation. Prophase. At the complete prophase, the chromosomal condensation, centriole poleward movement, disappearance of nuclear envelope, nucleolus, and Golgi bodies. Disappears, okay. Metaphase, we have metaphase, anaphase, telophase and cytokinesis. It starts with all the chromosomes scattered or placed observed throughout the cell. It starts with all the chromosomes scattered or least observed throughout the cell. Metaphase, it starts with all the chromosomes scattered or placed observed throughout the cell. Initial metaphase, the chromosome shortest and thickest. Best phase to observe morphology of chromosome structure. Centriole produce spindle fiber which connects to kinetochore. Centriole spindle fiber connects to kinetochore, and centriole produce spindle fiber which connects to kinetochore. Later metaphase, process of arrangement of chromosome in metaphasic plate, equatorial plate arrangement. The process of arrangement of chromosome in the metaphasic plate, congregation, anaphase, upstage. The separation of sister chromatid. Here, separation of sister chromatid towards the pole. Sister chromatids move towards the pole. Elongation of interzonal fiber and shortening of spindle fiber. Here, centromere divides. The separation of sister chromatid towards each pole. Separation of sister chromatid towards each pole. Separation of sister chromatids towards each pole. Separation of sister chromatid towards each pole. It's okay. Separation of sister chromatids towards each pole. Separation of sister chromatid towards each pole. Separation is brought by shortening of the spindle fiber. Separation of sister chromatid towards each pole. The separation of sister chromatid towards each pole. Connected to kinetochore and elongation of interzonal fiber. Sister chromatid towards each pole. Separation is brought about by shortening of spindle fiber connected to kinetochore and elongation of interzonal fibers. Centromere division towards the pole. Centromere division towards the pole. Chromosomes are trailing behind. Spindle. Interzonal fibers. Interzonal fiber. As each chromatids are separated. Transition to metaphase. Prophase, early prophase, late prophase. Anaphase, telophase, interphase, interphase, telophase, anaphase. Prophase I, metaphase I, anaphase. The chromatids are separated from chromosome. Each chromatids are separated. Each chromatids are separated from the chromosome. The chromatid aggregates at each fold becomes the part of dot nucleate. Chromatid is equals to 1 chromosome. Next, humans and 46. 46 chromatids. 2 chromatids are there. देखो पेंसिल चाहिए. This is pretty okay. Shut up, bro. Two chromatid, okay. Chromatid. 4 chromatid. 46 into 2. 92. 92 chromatids. Anaphase 46. 46 chromosome, okay. Anaphase, exactly same number of chromatid as Z2. G2 46 chromosome, 92 chromatids, exactly same number of chromatid as G2, double the number of chromosome as Z2. ये थोड़ा समझ में नहीं आया, मुझे फिर से करना पड़ेगा एनाफेज वाला क्वेश्चन. Telophase, reverse of prophase, chromosome chromatin, decondensation. Centriole disappears, spindle fiber disappears, nuclear membrane reappears. Centriole disappears, spindle fiber disappears. Actin and myosin filament starts pulling the cell wall. Formation of constriction towards the center, centripetal fashion, poro formation, okay. Mid body. Vesicles are produced from the Golgi bodies. The vesicles fuse to form phragmoplast. I don't want to think about it. Phragmoplast. Also, the ER vesicles contribute to phragmoplast. Okay. Phragmoplast precursor to cell plate. Towards the periphery. Centrifugate pattern. Centrifugate pattern leads result into calcium 2 plus and magnesium 2 plus pectate deposition, results in middle lamella, cell separation. Mitosis, auxin, cytokinin, zebraline, and one more is there, insulin. Encourage mitotic division. Colchicine, polyploidy. Metaphase I, the substance which suppresses half the division. Polyploidy, lilies. Azide, cyanide, mustard, yes. Agglutination. Growth and development of microorganisms, zygote, multicellular organism. Cell growth, okay. Maintains nucleoplasm growth of multicellular organism. Maintains nucleocytoplasmic ratio. Maintains nucleocytoplasmic ratio. Helps in maintaining, in case of growth of cell, helps in maintaining the proportionate component of both nucleocytoplasmic ratio. Maintains the surface area volume ratio. Surface area per volume ratio high. Cell repair and replacement. 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 Maintains chromosomal number. Maintaining the identity of chromosome. Maintains chromosomal number. Mitotic division maintains chromosomal number. Helps in repair and maintenance. Example, lining of the gut. Epithelial, outer layer of epithelium, lining of gut and replacement of blood cells. N is equals to N, number of times cell undergo division, number of times cell undergo division, that is N. N is equals to total time upon division of one cell, total time. Generation, okay, N is generation, total time upon and equal by one division. To the power n, where n is the number of generation, is equals to number of daughter cell, total number of daughter cell, to the power n, total number of daughter cell. Mitotic division, total number of mitotic division is equals to x minus 1, where x is the total number of daughter cells, total number of daughter cell minus 1, number of mitotic division. I'll go and make coffee for myself, okay? I'm kind of sleepy. I'll sleep otherwise. I think now my brother will also come, so I have to look after him. I think I'll go now, okay? Bye everyone. I'll go now.